Thank you for joining me for this next session of this lecture series. In this section, I am first going to talk about the scientific basis of this treatment process. Not everybody is interested in the basic science of surgical procedures. Some people would prefer to skip to the clinical applications that were more directly applicable to operating surgeons. However, it is critical to understand the actual biologic basis upon which these operations have been designed. Even though it is possible to achieve good results by simply following a specific protocol and surgical technique outline, to design new operations that have never been done before in a specific situation, it requires that one understands the basic biology that is operating and will determine the success and positive outcome of a proposed treatment. Therefore, I will begin by discussing the basic theory upon which this entire treatment system is based. Now, the basis of this system is to understand and appreciate the underlying method by which bone actually operates. In order to do this, we need to understand how bone was formed to begin with and have an understanding that the method by which the embryo used to form the skeletal systems to begin with still operates in the adult. This is not a universally accepted concept, and Dr. Egil Harvold, an orthodontist and anatomist, proposed this approach many years ago. Harvold believed that it was possible to reverse engineer the embryogenesis to understand how the embryo created skeletal systems to begin with, and then to utilize those pathways, that method, to create new operations to solve difficult and normal problems in the adult. Now, he had limited resources in which to figure out how this system worked. In fact, he was limited by simple observation. He was an expert in the treatment of craniofacial anomalies. So he was both interested in trying to figure out how to better treat these children, but also he was an observer that made connections between anatomical deficiencies and how they affected growth on children in general. So he also studied histologic sections of embryos to try to figure out how it was that the embryo was able to develop all of these structures seemingly without any external input. So Harvold's concepts were based upon the idea that a system to create skeletal parts was the product of many years of evolution. The attractiveness of this process was that the embryo is able to create complex skeletal systems seemingly de novo and without any external input. So the power of embryogenesis is one of self-assembly. In fact, the objective of all surgeons is to create a comprehensive and functional reconstruction without surgical incursion at all. One might think that this idea of creating a system to utilize embryologic processes is really not possible. And the first obstacle that we have to get over is that it is possible to understand this system, and that given a deliberate methodology, it is possible to create new operations that harness the system. Now, the embryo, as I said, has the unique ability of self-assembly. The embryo can create complex organ systems with no outside incursion. In addition, the embryo doesn't use input of external substrate. There are no grafts, there are no external input of any of the material that goes into making these structures. All of the material is created at the site by the embryo de novo. In addition to creating complex structures, these anatomical parts have to be able to not only develop, but to grow in real time, so that even the assembly of the basic architecture of these structures has to be redeveloped as the embryo grows. Now, the strategy to this is that there are challenges to create operations where there is no obvious solution to begin with. And in the past, we basically copy all of the operations that have been done by our predecessors and then reapply them to our patients in our clinical setting. The surgical design technique, which I'm going to describe now, is a major departure from the process which has been done in the past. The major strategy here is to create a system by which we can use newly discovered bone biology to design and execute operations that have never been done before. We are proposing to create a system of surgery that uses embryomimetic processes. The processes that the embryo uses to create the skeleton to begin with will be used to create treatments for adult patients. There is a logical pathway that this system must follow in order to be valid. The first obstacle is to define a specific mechanism by which the embryo creates these structures to begin with. This has been explored for many years, and there are many theories on this issue. In this series, we will focus on skeletal development. So the question is, can we propose a reasonable mechanism that the embryo uses to create a bone such as the mandible to begin with? In order to be valid, we need to have a process that can be verified with histology. If we can create a model, a theory by which the embryo used to create the skeleton to begin with, the next obstacle is to verify that this mechanism still remains active in the adult. Again, we need to define the mechanism and be able to demonstrate that the physiology and anatomy is present in the adult and working in an active way. Again, it requires histology to prove those objectives. If we can show that the adult has this mechanism in place, the next obstacle is to design surgical procedures that incorporate and exploit these mechanisms for the purpose of creating a solution to real clinical problems. The next step is to identify real clinical problems in our patients and then specifically and deliberately design new operations that incorporate these concepts. The fourth obstacle is to verify that if the reconstruction has been successful, that it remains stable long term. So even if we can show that, for instance, bone can be created by a system, it's still necessary to show and prove that the reconstruction remains stable long term. This means that we need to follow these patients for many years. So the objective would be to document successful reconstruction over a period of 5 to 20 years. Conventional surgery is a basic mechanical reconstruction. Conventional surgery intends to take a situation that is anatomically undesirable. For instance, a mandible which is of inadequate volume to achieve a cosmetic or functional reconstruction. Conventional surgery would follow a process by reconstructing that site by adding tissue, bone, or some bone substitute to make up for the volume that's missing. The idea being that the grafted or augmented bone piece will be incorporated into the living system of the bone and become a functional unit. The strategy here is to utilize the embryologic system to regenerate the bone and not require the inclusion of some sort of substitute material. Now, Harvold, in his research and observation, came up with a basic model on which he felt embryos use to create the skeleton. In this case, we see here that the mandible is formed in the embryo in a zone of soft tissue that follows very specific criterion. This zone in which the mandible appears in the embryo is adjacent to Meckel's cartilage. It isn't within Meckel's cartilage, it is actually at a distance from it. Harvold also observed that the zone in which the mandible forms is aligned with the axis of the mylohyoid muscle. So he concluded that these circumstances formed a situation where bone will form. So this is a outline or summary of Harvold's work, which showed that a bone will form when certain specific criterion are met. Harvold felt that a bone like the mandible will form if, number one, there is some source of cells in the region. Number two is that it required a zone of stabilization. In other words, the cells that would become mandible, undergo morphogenesis and organization, have to be in a volume that is mechanically stabilized. He felt that Meckel's cartilage, for instance, provided a mechanical stabilization in the soft tissue adjacent to it that produced an environment that was susceptible for morphogenesis if influenced by the correct signaling. So that would be the third requirement, that the cells in this special zone require some type of neuromuscular input. They require some type of signaling factor to be instructed to undergo morphogenesis and organization. In Harvold's theory, the signal would be coming through the mylohyoid muscle. He felt that the central nervous system created a signaling pattern that was delivered through the mylohyoid muscle into this zone in some sort of a mechanical way. The muscle obviously is a mechanical device, so he concluded that the signaling pattern must be delivered in some sort of mechanical or rhythmic contraction signaling mechanism into the zone, and then that was the signal that created forces that powered the process of morphogenesis. The fourth condition for bone formation is a lack of pathosis, a lack of pathology. Any type of infection or cystic formation at the site will prevent the formation of bone.